Oh, hello everyone. Thank you for joining us for this workshop. We will be focusing on the research on fermented feed for dairy cattle. And for in case you have not registered for the workshop, I would like to ask you to use the QR code on the screen and the right corner and to register so that we can have your contact information. And we are planning to send you a research summary we call fact sheet and after the workshop. And if you can register, so we have your email, we can send you the information. My name is Rui Hong Zhang. I'm professor in the Department of Biological and Agricultural Engineering at UC Davis. And today we have interdisciplinary team of experts. For this workshop, so I will serve as chair and MC for the workshop. And so I wanted to welcome you and also wanted to go over the program of this workshop and introduce speakers and also provide a little bit of background introduction for the project that we will be presenting and with the focus on fermented almond hulls for dairy feed. Okay, for today, we will start with the two presentations from Dr. Amias Kibro and Dr. Matthias Hess to give a quick overview of state of science related to dairy feed and nutrition and rumen fermentation. Then we have planned for three research presentations, which will present some highlighted results of the methods results we have from a project and we have been working on in the past 2.5 years. And then Nancy will come in to provide some final remarks. And I would like to ask you to hold the questions toward the end of presentation. So we will leave some time for Q&A and also discussion. Okay, and throughout the presentation, you're very welcome to post your questions in the chat, and I will monitor the questions, compile, and so that we can go over those questions. And certainly you are very welcome to also speak up and ask questions or provide comments when we finish our presentation. And before we go further, I'd like to let you know and wanted to conduct a mini survey and ask you to answer a few questions. And this one for now is what we call pre-survey. A few questions we'd like to have you to help answer. And so you can scan scan the QR code, the survey will pop up. And certainly, I believe there is a link in the chat now, and for you to use click the link, and the survey will bring up. So it will take probably a minute or so. We'll give you a couple minutes, and then we'll go further. Thank you and at the same time, also let you know the program of the workshop and also the bios of speakers and are in the document provided in the link of the chat. If you click the first link, and that will take you to the program and also speakers introduction. And so with a lot of details there, and also there's an abstract, research abstract in that document as well. And because of that, then I will try to stay very brief in my introduction of the speakers so that we can have time for presentations. Okay, Hamid, how we doing with the survey?
Yeah, thank you. I'm just looking at it now.
Okay, we'll move on. Thank you very much.
Yeah, only 5 responses. So
Hamid, you can clarify maybe that the survey remains open, so just leave that open for people who want to maybe complete it over the next few minutes.
yeah, it's actually in the chat now.
Okay.
Can you give 1 more minute, please? Just
Okay,
yeah, people
sure.
are still actually responding. So
I see. Perfect. Cool. Okay, well, thank you for completing survey. And we will next would like to introduce two speakers and to go provide the state of science overview and dairy feed and nutrition. And so first speaker is Professor Amias Kibro from Department Animal Science. And so second speaker is Professor Matthias Hess, also from Department Animal Science. With that, I will let them to speak.
Okay. I hope everybody can see my slides.
Yes.
Okay, so good afternoon, everybody. I'll be talking very briefly about dairy feed and nutrition, and just reviewing the role of almond hulls and byproducts. And so the main reason that we're using byproduct is because of its value, nutritional value, and California is the world's largest producer of almond hulls, or producer of almonds, and then obviously we have almond hulls coming from the production of almonds as well. And so because of that, we do have quite a large amount of almond hulls available in the state. And at the same time, California is the largest dairy state in the U.S. with close to 20% of the milk produced. So we have availability of almond hulls, and then we have a lot of dairy cattle in the state as well. And almond hulls and other byproducts will help in terms of managing the cost and reduce waste as well. Why do we want to use almond hulls? Well, almond hulls are rich in readily fermentable carbohydrates like sucrose and fructose and glucose, which makes them an excellent source of energy for dairy cows. They also provide fiber, which can partially replace traditional forages and contribute to rumen health. The soluble sugars in almond hulls are, as I said, readily fermentable, and because of that, they do not accumulate as much in lactic acid, and it prevents lactic acidosis, and it keeps cows from going off feed. And as a byproduct of almond harvesting, feeding almond hulls is a sustainable practice that reduces waste and can be cost effective as well, compared to other feed ingredients. Early studies show that fermented almond hulls may contribute to reducing methane emissions from dairy cows and offering an additional environmental benefit. So the almond industry, I'm sure, hopes that the feeding rate of almond hulls to dairy cows would increase in the future. But this can only happen if there is good and thorough understanding of the chemical composition of the almond hulls. This is a paper from Ed Peters and colleagues at UC Davis. So in the future, to feed high amounts of almond hulls to high producing dairy cows, nutritionists will need to be more closely scrutinize the chemical composition of almond hulls, and any kind of enhancement that can be done, whether it is through fermentation or others, would be quite important to provide the nutrition that the high producing dairy cows needed. And the FAO last year, or maybe a couple of years ago, they looked into the pathways to low emission future for livestock systems, and they looked at both in terms of the demand side and the supply side, and they showed that from the demand side, dietary changes, even if fully adopted, the potential is quite low, about 6%, but 70% of the reduction can be achieved from the supply side. And here we are really focusing on the feed and nutrition improvements. So adding more byproducts into the dairy system would help in reducing the environmental impact of dairying in general. And so using almond hulls as cattle feed really sort of diverts organic material from disposal or composting and integrates them into the food chain. So it promotes resource efficiency by turning what is a regional byproduct in California into a valuable input in livestock nutrition. And almond hulls are moderately, as I said, fermentable and are rich in pectins, which can favor propionate production over acetate production in the rumen. I'm sure Matthias will talk more about that, potentially reducing the methane formation per unit of feed. So this is what we're trying to look into this as well. And by using almond hulls, dairy systems can rely less on human edible crops. So there's the less human human food and animal feed kind of competition, and that will improve the sustainability profile of animal agriculture as well. So replacing conventional feeds like corn or soy and with byproducts like almond hulls could substantially lower the carbon footprint of the ration in general. Particularly if you look at if the transportation can be minimized, which is locally sourced, then you can really benefit from adding byproducts such as almond hulls. So I'm gonna stop here and then invite Matthias to continue this discussion.
All right, let me share my screen. Let's see.
Thank you, Amias.
Can you see the presentation?
Yes.
Or there's something else. Continue to share. There's something going on here. Not sure there's something in the middle of here. Do you see it? I have something showing up in the middle. But,
Yeah, we also see something in the middle.
and if I continue to share,
Yeah.
hold up, let me just, I don't know. I don't know why it's, huh.
I mean, you can see it, it's a
I
little
think
bit,
it
but
should be fine then.
yeah.
Matthias, do you want me to share? I have
If
your
you want to
presentation.
that's fine if you can share.
Yeah.
Yeah,
I think
you might want to try
I
it.
can try.
I cannot remove that box in the middle.
Yeah.
Maybe you have to make me the host in order to get rid of that box. But let's see if Royhan can. Really what drives Rumen function. And people who have met me, I think there's several of you in the audience who have talked to me before. By training, I'm a Rumen microbiologist or microbiologist specifically. And what we need to keep in mind when we have these feed additives or any ways where we want to change rumen function, what's really behind this is we want to change the microbes that live in the rumen, because all of those are really driving not only the function of the rumen specifically, but it also really generates methane, and we want to kind of change this towards less methane production and more towards volatile fatty acid production. Now, the challenge that we have with doing this more targeted, which is kind of what we would like to do, is the fact that, and you have seen that in the table that Amias was presenting, when you looked at the chemical composition of the different almond hulls that the Peters kind of collected from the literature, the chemical data from just the almond hulls was pretty much all over the place. And so we tried to address this more specifically by getting a better understanding of the chemical composition of all the feed additives, or specifically almond hulls for this particular project, so that we can be more specific about how we change the microbiome. And you can think about it really easily in the way of each of the different chemical components that are contributing to the overall makeup of your byproduct, specifically almond hulls. All of them have very different effects on the different components in the rumen microbiome. So we have bacteria, we have protozoa, fungi, and viruses, and all of those react differently to these chemical components. And then we also have the archaea. The archaea are basically related they're somewhere in between bacteria and fungi, and those are the methanogens. So they produce the methane. And so we try to be more specific at how we can change the function of the rumen microbiome. So Hamed, if you go to the next slide, please. And this is just basically highlighting again the value of having a rumen microbiome, and its importance for generating specifically the volatile fatty acids, which is then the product that the animal can take up. Now, unfortunately, in the process of generating volatile fatty acids, and Hamed, if you can go to the next slide, please. One more. I think I changed this exactly, is that what you can see here on the left, on the very left, is this is kind of a breakup, what happens to the feed. So on the left top, you basically see the feed, and you kind of can follow if you follow the arrows, you can follow the fate of your carbon and the hydrogen. And on the right upper hand side, what you see is basically what's happening to a large fraction, about 12% of the energy that the animal takes up is redirected into methane and it's being lost as energy and it's being released in the atmosphere. Now, ideally, what we want to do, we want to redirect the balance and we want to take the carbon and the hydrogen, and instead of having that go into methane and being released in the atmosphere, we want to go that specifically down what you see in the lower right corner. You want to kind of redirect it towards succinate and propionate, which then can be utilized by the animal. And so we are not only losing methane in the air, which is obviously not good for the climate, but we also want to redirect it for the gain of additional energy so that we can utilize that towards more protein production. Now, what you saw on the previous slide, that was, and Hamid, if you just jump back, this is basically a representative animal that has these fistulas. And so we have a couple of these different animals in the dairy, in the teaching facility here at UC Davis. And what that allows us to do, or specifically what this allows my group to do, we can go into the dairy and we can collect rumen content and then bring it to the laboratory. And then we can start testing different byproducts, and here specifically our almond hulls. We can ferment the almond hulls, and we can then test what the addition of these byproducts, what the effect is on the rumen function. And Hamid, if you jump forward 2 slides, please. This is just an example of the different in vitro rumen fermentation systems we have in the laboratory. So we have a couple of more, which I don't really show here, but the idea is the same for all of these different systems, is that we can go to the dairy, we can collect rumen content, including feed and including these microbes, and we can then basically inoculate the different fermentation systems. So you see that in the middle, that is the system that we used for this particular study. And what that allows us to do is to evaluate how different treatments, so treatment meaning, go back, please. Ahmed, yeah, thank you. So treatment means we can have almond hulls fermented in different ways, and then we can incubate that in a water bath, simulating or maintaining the same temperature that what we would see in the rumen. And at the same time, while we are collecting, or while we let the feed ferment, we also collect gas, which will be collected in these silver bags. Thanks, Ahmed. And we can collect gas. After we collect the gas, we then can take these bags, we can then remove them, and we can take them into towards in the other lab where we have a gas chromatograph, and then we can evaluate how much, not only how much gas is produced, but also how much different gas components. So specifically, we can look at carbon dioxide, hydrogen, and most importantly, methane production. And then we can evaluate really how much reduction ideally we'll see based on a different treatment. All right, I think if you go one more to the next slide, this is just kind of the ideal scenario, which is a study we performed with Amias a couple of years ago. And this is kind of the gold standard. But reason why I want to show you this is kind of the system basically allows us to identify potential winners we would like to take to the market. And what you see here is kind of what we found for red seaweed, which most likely all of you have heard about. But the idea here is really that once we find something that's more sustainable for California, such as almond hulls, we can actually then go back to Amias, and we can then perform animal tests. And so that really allows us to take many different pre-fermentation conditions to find winners that are competitive with the red seaweed, since this is not something that is readily available to the entire industry. Okay, this I think is all we have for kind of the fermentation introduction, and I'll present results from the almond hull study in the second part. Thanks, Hamid.
Thank you very much, Matthias. I will share my screen. Okay. Okay, so I wanted to tell you about a little bit the journey of our fermented feed research. And this team of our team of researchers here started to work on fermented feed back in 2020. So we had exploratory grant from Almond Board of California, and to look at the effects of almond hulls on reduction enteric methane emission in the dairy cattle. And so that was a 1-year study. And from that study, our results showed that there was a significant enteric methane emission reduction when we we did the in-vitro study, in-vitro testing, and then showed the significant methane reduction. And so now we had the follow-up project funded by California Department of Agriculture, Food and Agriculture, CDFA, specialty crop grant. And so that is a 2.5 years project. And so it will conclude the end of this month. And so and it's the title is Creating Nutritious, Highly Digestible Animal Feed from Almond hulls and the Tomato Pumice. So we continued to study, research the fermentation of almond hulls. We also added the tomato pumice in the mix of substrate material we ferment. So we are at the end of this project, and that's where we're here today. Wanted to share with you some of the results from this project, CDFA project. Also, recently, we started another continuation of project funded by California Dairy Research Foundation. And so with the PI, Matthias is the PI for the third project. And so it is called the fermented agricultural byproducts as a dietary modulators to reduce enteric methane emission from dairy cows in California. And so it's a fabulous project. Okay, so the names are fabulous. And so that's a kind of looking to the future. We started this year, and it's a great continuation. So I wanted to thank all the sponsors of our research, California Almond Board, California Dairy Department of Food and Agriculture, and California Dairy Research Foundation for supporting our research. Why fermented feed? Okay, so that is a good question. And so we wanted to look into and fermented feed. We as a humans, so we eat the fermented food all the time, right? And whether it's beverages or solid fermented food products. And the health benefits of fermented food is widely recognized for humans. And how about for animals? The food fermented feed for animals. And so there has been, much less than for humans. And so fermented feed is not widely used in animal industry. And however, from research standpoint, fermented feed has been more studied for pigs and chickens, okay, swine, poultry. And so it has documented in the literature and some testimonials and data from industry, and have showed the benefits of fermented feed for animals. In this case, more about for chickens and pigs. Improve the performance and nutrient digestion in pigs, and reducing antibiotic use in pigs, and also decrease mortality rates, improve immune responses in chickens. And so also last few years, I have seen a lot more research in the industry adoption in Asian countries, especially in China, for fermented feed. Okay, but here back in the U.S., and we're more limited use or study on the fermented feed. So just looking at the literature, very few studies in the past have been focusing on cattle fermented feed. Okay, and so however, some research documented the probiotic fermentation with the yeast shows nutritional benefits, a potential reduction in methane emissions. And so we have, in our research in the past few years, so we have focused on fermentation of almond hulls. As Matthias and Amias laid out, and almond hulls is a byproduct of almond industry and a huge quantity, and so has a good amount of sugar. About 20–30% of almond hulls dry basis is a sugar, and also good dietary fibers for the cattle, and also has a 4–5% of protein on dry basis. But in addition to those big components, and almond hulls is very special. It has many types of phenolic compounds, which are which have antioxidant properties, and there are a few listed in this slide here. So almond hulls, in addition to just as a feed, the supplement for calories, and it has compounds that could potentially offer a lot of health benefits, special health benefits for cows, for cattle, and for other animals as well. Okay, so, and in the following presentation, we're going to focusing on the project that we have been working on the past 2.5 years, sponsored by CDFA. And so it's called Creating Nutritious and Highly Digested Fermented Animal Feed from Almond hulls and Tomato Pumice. And we'll present a little bit the data on the tomato pumice, but mostly our presentation will be on the almond hulls. And again, I want to thank CDFA for funding this research. And also the principal investigators are myself and Hamid, Matthias, Amias, and also John Lee Pan from Biological Neg Engineering. And so we have involved many students, staff, and so including graduate students and undergraduates in this project. So I want to thank them. The research objectives are mainly these four shown on the slide. The first one is develop optimize fermentation process for almond hulls and tomato pumice. And we also looked at the tomato plant biomass. And the second objective, evaluate the quality, nutritional value of fermented feed using in vitro and in situ tests. And the third one is conduct on-farm fermentation of almond hulls and the tomato pumice. And the fourth one is determine the emission of methane from cattle fed on the fermented feeds we produced. So next, I would like to introduce three speakers who will provide detailed presentation for the research we have conducted on this project. And Dr. Hamid Yelmashet from Biological Engineering, and so Dr. Hess from Animal Science Department, and Dr. Afroza Akita, and she's postdoc in Animal Science Department. So they all have them to share their presentation. Okay, so Hamid first.
Okay. Can you see my slides, please?
Yes.
Okay. Yeah, good afternoon, everyone. I'm going actually to go through the first two objectives that presented by Professor Jones. That is actually optimization of the fermentation process and scaling up and producing enough fermented feed for the animal studies that conducted by Professor Kibriab and Afroza, and also produce some material for Dr. Hess to test in his in vitro. So the first actual experiment that I would like to present here that we did is producing the fermented feed from almond hulls and tomato pomace. And here we actually used almond hulls that we collected from a processor, and this almond hulls were ground using a food processor. So as you can see, actually particle size may be about half centimeter. So basically we screened actually the material, and we used the particles below 4.75 millimeter. Similar, actually, we get also tomato pomace from a processor here in California, and we screen them also with the same screen size to produce also the same particle size that we used in this experiment. There are some fibrous actually materials actually larger than 4.7. We excluded them actually that for consistency of the material. As we can see here, this is just the composition, and the almond hulls that we used in this experiment was independent variety. It has a high sugar content and total solid about 85% and volatile solids about 85% of the total solids. Tomato pomace also has high solid content and higher VS in fact. Also, tomato debris, this basically is what we excluded, still also has VS higher than the almond hulls, but still has a lot of fibrous material. So we only concentrated on almond hulls and tomato pomace in this experiment. So what we did here, we actually, based on the experience from the project funded by Almond Board, we actually selected some conditions actually to produce this feed. The fixed parameters here are a temperature of 35 degrees C, and yeast loading was 2 grams per yeast per kilogram of feedstock, and the moisture content was 70% of wet basis. And the fermentation was 14 days. And again, this is basically, we compared the almond hulls alone and the tomato pomace, and the 50-50 of almond hulls and the tomato pomace. We measured the yield of volatile fatty acids, especially actually acetic acid in this case, and also ethanol and lactic acid. So to prepare this material, we used actually the Saccharomyces yeast, which actually this is basically bakery yeast that is actually over the shelf here in Costco, and we had to replicate of each material, and of course we have to prepare them as the yeast and activated it, and then we used the vacuum bags to as containers for fermentation. And vacuum bags means that we don't have any air bags during the fermentation. So after 2 weeks, we measured the lactic acid, acetic acid, and ethanol. As you can see here, this is the control, means that there is no inoculum. So this is the hulls, and this is the pomace, and this is the mix. So if we look at this one, we can find basically, if we have pomace, we have a little bit of lactic acid compared with the hulls. However, actually, if we mix them together, actually jump it to high concentration of lactic acid. And also here, the acetic acid increased as well, and the ethanol also. However, ethanol here in the mix was lower than the hulls alone. However, if we have this yeast added, this is a treatment, basically we increase the ethanol production and the acetic acid compared with the control, mainly for every of this treatment. So we also, one of the objective here to, because the almond actually industry moving toward actually off ground, harvesting, we compared also the holes that collected on ground and off ground. Similar actually conditions, 35 degrees C and 70% moisture content and 2 weeks. We compared also the on ground and off ground, and we measured the same thing actually, ethanol, lactic acid, and acetic acid. So if we look at this also figure here, we can find actually on ground without actually yeast, and off ground also without yeast. Basically, this was actually surprising that actually adding the yeast actually didn't improve the or increase the ethanol production, probably also because the off ground was basically doesn't have any indigenous microbes there, and also it was actually free of lactic acid, in fact. So this means that actually the holes alone, if we actually collect it from the ground, it might be actually having some kind of contamination from the ground. And this actually, I will come to this one of the recommendation of the research later on. So basically, this might be one of the factor actually we see some lactic acid in these cases. So again, if we have also off ground, basically compared with the on ground, basically the on ground also produce higher ethanol, higher acetic acid, and higher lactic acid. Again, the pH also we measured at the end, basically the on ground and off ground, basically adding the yeast, of course, it's actually decreased the pH. So we came actually also because a lot of actually parameter can actually affect this kind of intermediate or basically the component that we measured. So we have to optimize the conditions for this fermentation. And here we actually also have 2 weeks. The loading is basically the amount of the material that we used in the bags, the vacuum bags. So we studied actually the effect of different temperatures, again for 25, 35, 45, and then yeast loading also 0, 5, 10, and different moisture content. As we can see here, basically, this is actually a little bit crowded because of the many actually data points here. But if we look at this one, we can say actually that the moisture content at 70% can be actually recommended, and the temperature of 25 degrees C to 35 degrees C. So basically 25 degrees C here, and maybe 10% moisture content and 70% 10% yeast and 70% moisture content can actually produce a high ethanol and high lactic acid and acetic acid. And also, again, here this also for 70 degrees C, also the same, more or less actually the same values if you look at this kind of bars here. For that, actually, we found actually this range of temperature may be optimal from 25 to 35 degrees C, and for 5 to 10 gram of yeast per kilogram of the holes. Again, the moisture content is 70%. So basically, this is a little bit higher than the off ground. The off ground about 60%. So basically, 70% if actually off ground is an option, still actually maybe need a little bit of water if we need to modify the moisture content for this fermentation trials or for large application, basically. So we came also to the point that, okay, we need actually to have this material to be delivered to animal study. So we have to see also what would be the scaling up from this small vacuum bags, what would be the condition or basically what would be the characteristics of the feed that can be produced if we scaled up this method. So basically we use the same actually almond holes also that we collected from the processor, and we hammer mill them. We used the same particle size, and we selected actually 25 degrees C as conservative in terms of temperature. So basically, at that time, we didn't actually think that adding also heat might be actually a cost added to the process economics if we have to scale this up. So basically, we selected also 25 degrees, which is one of the middle point actually, or one of the points that we tried in the optimization experiment. So again, 70% and 5 gram of yeast. So we used actually duplicate in this case to 5-gallon buckets. and inside the incubator at 25 degrees C, and also we pass actually the off gas from these containers through gas meter, and also this basically a water bath to account for the gas, and also for basically prevent any leak of air to the heat space of the reactors. So as we can see, actually, this is basically at the beginning, at the end, this is very beautiful, actually, surface of the material. There is no mold, there is nothing. So basically, one of our concern actually was about the mold, actually, for during this fermentation. Again, here, this is basically at day 0 and at day 14. And a little bit, actually, moisture content increased, of course, because of the reduction of the fermentation. Basically, some of the solids actually converted to maybe carbon dioxide during this process, and also some intermediate like volatile. Basically, this also can be evaporated during the moisture content actually determination. As we can see here, this is basically very comparable to what we have in the small bags. We have actually ethanol about 0.12 gram per gram feed, and acetic acid 0.7 gram per gram feed, and lactic acid 0.8 gram per gram feed. So one of the other question also about this, what would be the stability of this fermentation? Because basically we can't actually expect that the farmer can produce it today and use it actually after 14 days directly. So basically what we did, we kept actually these 2 buckets for 8 more weeks inside the incubator. So what we actually also over the 8 more weeks, we measured actually also the lactic acid, acetic acid, and ethanol over time, and also pH. The pH was very stable, but here actually surprising is that the lactic acid uh, actually reduced by about 50% over the storage time, or basically the stability time. And also the ethanol was more or less actually stable, a little bit maybe about 5% or 10% reduction. And acetic acid also was about maybe also 10% reduction. However, actually, this again, the stable pH. So this means that maybe also we could maybe recommend that the material can be used maybe as soon as possible, but still actually there is nothing in terms of mold or anything. Just we can keep in mind when we scale this up, if we need actually to have storage, we have to be careful about this kind of intermediate change over the storage time. So next actually step, we're happy with this result, and then we moved actually to farm scale fermentation. And to do this, we have to get a large amount of holes basically from a commercial vendor actually for this almond holes. So we get actually this from one of the companies that provide almond holes for dairies, and they deliver this material into big sacks. We get about 5 tons of material, and this is the composition here. So basically the moisture content was 9.6, and protein about 6.8, and fat was about 3.9. And surprisingly, actually, that these holes had little sugars compared with what we had into the small scale experiments. So here, basically for 10%, which actually in line with what Professor Kibriab presented in his table, one of the studies also reported actually this kind of 14%. At that time, we asked the vendor, he doesn't know actually exactly the variety. He said that it's actually mixed varieties, maybe. This is why maybe actually one of the things that we had in this one. So the first actual experiment for this scaling up, we actually went from 5 gallon to 35 gallons. Basically, the first actual experiment was a pilot scale. This will be Afruza in next talk about if the cows actually will like this or not. Because basically you can produce anything, but maybe the cows will actually will not like it. So basically what we did actually this experiment basically to scale this up as the first, almost actually similar condition, except that this has been conducted into a greenhouse. at a set point of 30 degrees C. So basically, this was all the greenhouse that might have actually some temperature fluctuation. I will actually show a temperature profile later on. So what we did actually, we get the yeast, the same kind of yeast, we activated them. We used actually a concrete mixer to mix the material with the yeast and the required amount of water to adjust the moisture content to 70% moisture content. And then we filled actually this kind of garbage buckets. So here actually we kept the material, we kept this buckets into the greenhouse for about 2 weeks. For actually not about, it's actually 2 weeks exactly. And then we sampled actually. Surprisingly, actually after 2 weeks, we found some kind of molding in the surface of this buckets. Mainly because actually we were actually insulating them with some kind of also air venting and all of that, but because of actually the sealing of this kind of bucket was not really actually fully sealed, so basically air penetrated and it has some kind of small layer of mold. So basically what we did, we removed actually this mold out, and after we removed the mold, actually underneath it, it was actually beautiful also feed with this kind of composition here. Lactic acid was 0.07, and the acetic acid was 0.05, and ethanol was 0.04. So basically, this concentration was much less actually than what we had in the lab experiment, mainly because of um, the whole type, the holes type. So from that, we actually had to change the buckets to other buckets with more sealed. Actually, this kind of clamp, kind of uh, of sealing, and we also used the same actually process. We also activated the yeast. We used the same mixer, the concrete mixer, and then we produce actually this basically under the same condition, the same greenhouse. We produced about 12 batches, each was about 900 kilograms. Basically over the 12 months, 12 weeks of delivery to the animal study, we produced about 11 ton of the wet material. Oh, this is basically the concrete mixer. So basically here, this is the materials here inside the greenhouse. And we also have this kind of actually water trap, basically to prevent any air to that come to the head space of this kind of well-sealed drums. And here, this is basically the delivery for these drums to the dairy at UC Davis. So during this actually 12 weeks, we did actually take samples, compose a sample of the whatever delivered to the animal study, and we measured the lactic acid, and we measured also ethanol, and we measured acetic acid. Surprisingly, actually, the ethanol was more or less constant, and also acetic acid was also more or less constant. However, lactic acid was reduced basically here. So from week 8 until the end, there was some reduction, mainly because actually the temperature inside the greenhouse was reduced actually from about 30 degrees C to about 21 to 20 to 21 degrees C, basically about 60 degree Fahrenheit. The pH actually was almost constant, about 4.5. So basically, I think the feed was actually good for the cows, and they liked it. And Fruza will actually present this about the effect of this feed on the performance of the cows and the emissions. So besides that, we did also scale up the tomato pumice, actually similar to what we did also for the holes. Basically here, more or less the same actually, like also lower ethanol and lower acetic acid and the lower lactic acid in this tomato pumice. Again, there is no actually problem with the the mold or anything, just a beautiful surface and high quality, very pleasant actually smell. So the conclusion actually here is that almond hulls fermented better than tomato pomace, of course, because of the high sugars. Even with this kind of lower sugar concentration in the hulls, still actually producing a little bit higher fermentation, actually intermediate like ethanol, lactic acid, and acetic acid. And we can recommend actually maybe 14 days fermentation and 25 to 35 degrees C at moisture content of 70% moisture content and yeast of 5 gram per kilogram. And the lactic acid decreased actually by about 50% over 8 weeks of storage, but ethanol is still actually, and acetic acid was more stable beside actually the pH. Again, the last conclusion that the fermented hulls produced at the farm scale had lower concentration of acetic acid, lactic acid, and ethanol compared with the laboratory experiment, mainly because of the type of the hulls, probably a little bit of the temperature, but actually mainly I think because of the sugar content in the hulls. What we need actually to do next actually for future research. So basically we need to do more on farm scale fermentation using hulls from different sources at more stable temperature. Maybe we need actually to get a better greenhouse and conduct also on farm fermentation for co-fermenting almond hulls and other byproducts. Maybe also still maybe good to have tomato pomace or other materials. And we need also to study the effect of byproduct sterilization or pasteurization. Basically, this is important because as we could see from the off-ground almond hulls that has actually different compound basically and as intermediate, mainly because of actually the contamination of the almond hulls from maybe the indigenous actually microbes that can actually be caught from the ground while actually harvesting. For sequential also production of feed, maybe we can study also the recycling of part of the fermented feed as inoculum, because basically all of this experiment has been done with fresh yeast. We actually think that if we can take part of this fermentation of fermented feed as inoculum for next batches, might be also save the cost for yeast. With that, I would like to thank you.
Thank you, Hamed. And so our next speaker is Professor Hess.
Okay, let's see if the sharing works this time.
Yes.
Okay, here we go.
Oh, sorry.
That's okay. Now it worked.
Okay, perfect.
Okay. So I'm going to talk just briefly about the in vitro portion. And just remember this is kind of the initial step that led us to identify really here are the fermentation conditions that we are using to generate the fermentates that are most likely having the effect that we would like to see in the animal. And so Hamed and Professor Sang, they were kind of fermenting almond hulls and tomato pomace at very, very different conditions, various different conditions. And we then took these these fermentates and evaluated them in vitro. Now, I'm not going to show any of the results because there were many, many runs that we performed without success, meaning we actually had fermentates that did not reduce methane. Those are the ones we are not showing, but that's really the value of that in vitro system, is that we can test many different varieties in relatively short time without having to run our animal studies. Now, for the experimental design, what I would like you to understand is pointer options. Here we go. Is that we took fermented byproducts. So in this particular case, I will show you results for the fermented almond hulls. We then inoculate our fermenters or our small bioreactors with rumen content, and then we add small nylon bags that contain feed. I show you that on the next slide. We put them into these bottles, and then we measure gas production over a total of 4 days, so 96 hours. And we'll collect then gas specifically. So we collect gas over the entire time, but we have to take specific measurements since we have to disconnect the gas bag, and we'll do this every 2 days, every 48 hours. And then we actually carry the gas bag to the gas chromatograph, and then we can evaluate the gas profile. Now, what's important to understand is that when we disconnect the gas bag, we also add new feedbacks, and so that happens every 48 hours. And this we try to mimic the feeding process. Obviously, we can't feed them, or we can't exchange the feedbacks every 12 hours or 24 hours because we would disrupt the anaerobic conditions. So the 48 hours to 2 days is kind of an intermediate to make sure that the anaerobic conditions are maintained at least for 48 hours. And that's really important because the microbes in the rumen are extremely sensitive to oxygen. And so this gives them the ability to kind of get reactivated, and so allow them to really perform the way they would actually perform under native conditions. Now, what you can see here, those are the unfermented almond hulls, and those are the fermented almond hulls. So physically, you don't see a difference, but the chemical composition actually has changed over time, as Hamid was kind of pointing out. And here are these nylon bags. So they're like little tea bags. I mean, pretty much they are same size, but the difference is that they don't contain tea, but we have total mixed ration, and then we actually add the different treatments. So either we have no treatment, or we have the red seaweed as a positive control, or then we have the fermented almond hulls in different concentrations, or unfermented almond hulls. Now, if you kind of look at this in more detail, so this is the experimental setup. We run every treatment. We take rumen fluid. Let's start here on the left. We have 2 animals from which we collect rumen content. We mix that in the laboratory. We mix that together to get a better representation of the biological diversity that we actually see in different animals, since all of that can have an impact on rumen response. We then take our regular total mixed ration that is fed to the dairy animals, and then we actually have a saliva buffer, which allows us to maintain the pH over 4 days. Just to think about it, the microbes usually they generate the volatile fatty acids. So what you would see usually if you don't have a buffer, you would see a drop in pH, which then would be problematic because that would be acidic, and we want to avoid this, and that's we can do this by adding the saliva buffer. So this allows us to maintain stable pH, which I'll show you actually with data. The different treatments that we use besides the negative control and the positive control just allowing us to show that, ensure that our system is actually working properly, and gives us also kind of a benchmark for where we would like to go. And then we have fermented almond hulls at 5% inclusion rate and 10% inclusion rate. And then we have unfermented almond hulls at 5 and 10%. And then we basically to put that into our in vitro systems, incubate them at 39 degrees Celsius, and take samples every 48 hours. And what we found here, so we basically what I show you here is on the left are the graphs. So if you're a little bit visually more attracted, then you can actually look at the graphs. If you want to have details, you can actually look at the table on the right side. What's important here, though, the first group here are the results of, or the amount of methane we have been measuring after 48 hours, and the second group here on the right is after 96 hours. First bar is always the control, so untreated total mixed ration that we incubated in our in vitro systems, and you can see how methane is being produced over time. So if you just look at the negative control over the two different time points, you can see that methane is increasing, which is kind of what you would expect. Now, the second portion, and so the asterisks then also indicate significant reduction, and that we see a significant reduction with the red seaweed, which is not surprising, but that also shows us that our system is working properly. Now, important is the second groups here, the second gray bars that you actually can see. Those are fermented almond hulls that we added at 5 and then 10%. The light green bar is 10%, and you can see already after 48 hours, we see a significant reduction in methane. If you want to see the numbers, you just simply go to the right, so you actually have the quantitative amount. So 5% of fermented almond hulls reduced methane by about 18%, and at 10% we saw a reduction about 34%. Now, after 96 hours, we also see a significant reduction with 5%, and a similar reduction. So most likely what that indicates that when we add fermented almond hulls at 10%, the maximum reduction is already being reached after 48 hours. And so there's no further reduction, even if you feed it longer. So we don't know necessarily what's going on there. That's kind of a microbiome question, but this is really promising. You can also see here, if I just show you on the table, because those are the real data, I think that's more interesting than looking at the graphs, is that we actually also can reduce methane production by simply adding almond hulls, but there's no statistic significance there from the measurements that we took. So when you really want to be sure that you can actually reduce methane, then you should really pre-ferment your almond hulls as we have done in our animal trial. Now, we can also measure total gas production, and this kind of just gives you a little bit of an idea about fermentability. And what's happening here is, and that's a little of a downside here maybe, is that when we ferment our almond hulls and we add that to our feed, it seems like it's slightly less digestible, or but I think the question, and don't forget, this is all in vitro. So for complex mechanisms such as digestibility, the animal study will be really providing the real answer. So there are downsides to utilizing in vitro systems. That is clear. And what's important here also, I touched on this briefly, was pH. And what you can see here, so here we have our control, and you can see the regular pH here was 6.7 after we started the inoculation, and we pretty much maintained a relatively stable pH, regardless if they were fermented or unfermented after 48 hours or 96 hours. So that remains relatively stable. So with that, and again, like the other, we did many other preliminary tests here, but none of the other fermentates produced any reduction in methane. So that's the reason why we don't show you the exact same graphs without any changes in methane reduction. So with that, I actually want to hand it over to Dr. Akhter, and thanks for listening.
Thank you very much.
You can see my screen, right? Okay.
Yes.
Okay, good afternoon, everyone. I think by this time I will have an idea what is the nutritional composition of almond hulls and why California, and what is the effects if we ferment them. And you all can see my screen, right?
Yes.
Yes.
Okay, now here. So basically, from our part, we did the on-farm testing of fermented almond hulls, and we see the effect on almond hulls on milk production and methane emission in dairy cows. So I'm gonna talk about most of the results, what we have observed in here. I'm trying to access it. So yeah, I'm trying to find the next option from my slides, and looks like I'm having issues with going to the next, and for some reason it's not working.
Would you like us to share?
Let me try one more time. Maybe that would be great if you can do that, Hamid. And I'm going to stop it. Thank you, Hamid. If you go on the, yes, so I'm not talking about what's on the background, and I'm sure you all have a great idea how we can have like better effect if we ferment them, and fermenting them could mitigate methane emission in dairy cattle based on some of the in vitro stuff that Dr. Hess pointed. And why we need to do that study on farm, because there is limited research on the effect of feeding fermented almond hulls on the production parameters as well as the enteric emissions in dairy cattle. If you go to the next, Hamid. So we already had an idea that the effect of in vitro results that we can have from fermented almond hulls, and we already were working with Dr. Zhang's lab for getting a big bulk of fermented feed so that we can feed to the cows and see how they're doing, and with their in terms of their production and enteric emissions. From our study, we included two inclusion rate, that was 5% and 10% on dry matter basis, and we hypothesized that including fermented almond hulls at 5 and 10% of dry matter would significantly reduce enteric methane emission while improving milk yield. And the main objective of our study was to investigate the effects of supplementing fermented almond hulls in the diet of lactating dairy cows on enteric methane emissions, milk production, and milk composition. So before we plan a study, we designed a pilot study because the farm people were like a little bit concerned if the cows will be liking them and how the cows will be doing. So we wanted to test the feed on cows. So we selected 2 cows, and as we were planning to include 2 inclusion rates, we wanted to see if we provide 5% and 10%, how the cows will be doing. So as usual, like how we do it on the farm, we were feeding them in the morning and evening, basically like 65% in the morning and 35% in the evening. And they were fed for 1 week. After the end of 1 week, we saw that the average intake was 35.2 and no adverse health effect was like found. And as I was monitoring the cows, we did not see like anything that was notable, and we concluded that yeah, there was no effects like adverse effects. So we wanted to proceed with the further study in a big scale. And the main dairy trial that took place in last year from August to December. To design that study, we started with 24 dairy cows. But first of all, I would like to point some of the chemical composition of the trial ration. And as you can see, we included alfalfa hay, wheat hay, almond hulls in only the control group, and whole cottonseed, grain mix, and some other nutritional ingredients, as well as the fermented almond hulls in only 5% and 10% almond hulls group. And then we also looked at, if you go to the next slide, Hamid, we also looked at the composition of fermented almond hulls. Basically, we from different weeks, we composited samples, and we submitted for analysis. We saw that the dry matter percentage was about 25%. The CP was 9.7%, and we also observed the lignin was 20.8. As well as if we, when we had all of those things set up, we proceeded with the main study. If you please go to the next slide, Hamid. Yeah, first of all, we took 24 cows. For that, we took mainly Holstein cows. They were almost 100 days in milk and 636 kilogram body weight. For that, we utilized the electronic Callan gauge. So at UC Davis dairy cattle facility, we have some feeding opportunities with electronic system where individual animal can access their own bunks, so that we have an idea of how much they're eating each day. And let's say on the next day, if they're from their oats or their whatever the leftover they have, we can understand if they're eating less or if they need more. So we trained them for 2 weeks. Cows are very smart, so they learned it very quick. Maybe within 1 day or 2 days, they knew how to go to their own bunk, and they were eating well. But we wanted to give them enough time so that they know how to utilize fully their bunks, as well as the green feed machine that I'm going to talk next. And then if you go to the next slide, Hamid. For the study design, I would say we had 2 weeks of training period, and then as soon as we knew all the cows are well trained, and we moved to the control ration. That means all the 24 cows were receiving control ration for 2 weeks, just to make sure that we're well adapted to the rations. And the study design was Latin square design, which means the study had 3 different treatments and 3 different time periods, and each time period consisted of 4 weeks. That means for 4 weeks, 8 of the cows received 1 single ration, and then after 4 weeks, we switched their ration, and then again after 4 weeks, we again switch their rations. So if you consider on this pattern, that means all the 24 cows received all the ration at any time points. And then if you go next, please, Hamid. So we were following the farm procedure for feeding and milking, and for the health maintaining and health monitoring everything. So we were feeding their morning and evening, and we only collected the refusals in the morning, and from the refusals we knew what was their daily intake, and then we had an Excel sheet that automatically calculate how much they are going to need for the whole day. And we divided the ration for the morning 65%, and then evening 35%. So we weighted how much individual cows are gonna need, and then we also weighted the fermented almond hulls, and then they were mixed manually. They were provided at their bunks. If you go to the next slide, Hamid. We collected their daily meal yield, their feed intake, how much they were eating, and then each day we knew like if their intake is going low, and if that's like too much, we had to inform the dairy, and if they're like eating too less for 2 or 3 days, they were checked by the vet. We also had their weekly body weight. We also had their milk samples for analyzing fat percent, protein, lactose, SNF, milk urea nitrogen, and somatic cell counts. And they were also monitored for health, and especially they had any digestive disorders as we were feeding almond hulls, and then after 4 weeks we switched. So especially we wanted to make sure that the change of the diet were like making any upset to their digestive system. And for enteric emission collection, we had large animal grain feed system from CLOC. From that machine, there also went a little bit of training process. As I mentioned before, that during the training period, we made sure that they were trained on how to use the individual bunks. They knew that this is my bunk. And likewise, they also knew that the machine was dispensing some feed for them. So that machine can capture enteric emissions from their breath samples. So we program the systems in that way so that the animal knows they have to visit the system after several hours, and then they will be getting some of the sweet treat. We provided them some alfalfa pellets. So they knew that I had to visit the system after several hours, and I will be getting some sweet treat for that. And then when they were eating on the system here, their breath sample went into the system, and that's how the machine can capture their enteric emission. And by the end of the study, basically the company CLOC, they processed all the data, and then we had the final data, how much each of the methane, CO2, and hydrogen was emitting by individual cows. If you please go to the next slide, looking at some of the results, we observed that there was no difference in dry matter intake based on their treatment. That means control 5% and 10%, all the group were eating more or less the same, and then they were not significantly different from each other. And then likewise, body weight weren't different based on different treatment groups. And
Thank you very much. Afroza, can you bring your slides back up on the conclusion? Oh, okay, Hamid, so you're operating the slide, if you can bring that up. So we would like to open up for questions, comments, and I think we're good with our timing. And so if we can, have our audience to post any questions or provide the comments, and afterwards, and then myself, Matthias, Mias, three of us can offer some final words. And I will also, before you leave, and we like to ask for a favor again and asking you to do a mini survey. Okay. But with that, please, if you have questions, and then you can unmute yourself, ask, or if you wanted to type your question in chat, then I'll be happy to ask for you. Any questions from our audience or comments from our audience?
Yes, this is Paul Souza with Western United Dairies. Thank you very much. Great presentation and really impressive work that you guys are doing. My question relates to the base diet and the results that you're seeing from that. I see the base diet had wheat hay, alfalfa hay, almond hulls, whole cottonseed, and then a mineral mix and salt, but it did not include fermented corn silage, which is very typically fed in the San Joaquin Valley to dairy cows. And so what my question is, would the results be different, or are we getting those benefits already from fermented corn silage? And that would the fermented almond hulls add to that? And you know, in moving forward, if you're going to do additional work, I would suggest using fermented corn silage in the base diet to compare that to, you know, more commonly fed rations in the Central Valley. Thank you.
Yeah, this is Stan Weller. I would absolutely agree with everything that was just said. Not just for California too, as moving abroad. I mean, you're outwardly from California. Corn silage is a very common feed, and so I think that a lot of dairies, both in and out of the state, are using it. And so I think it definitely would make sense to evaluate that. Thanks.
Thank you for that feedback, Paul and Dan. Yes, I think if you had more corn silage in the mix, we probably see some differences there. But still, at the 10% level, it was quite similar, which is unusually when you look at the in vitro and in vivo results, they kind of mirror each other. So you don't always see that, but I think we have fairly good confidence that at that level, we will be able to see some benefits of including almond hulls and for the reduction of emissions. And this is just direct emissions as well. Well, the other thing that we can do is looking into the whole system and the whole ration and what is the environmental cost of the whole ration, and then we can compare at that level, and we could see the differences in the carbon footprint of the rations that we're using here or in the Central Valley, and then when we add the 10%, there will be an additional advantage in there as well.
Thank you.
Hi, Ray Hong. This is Guang Wei. I have a couple of questions.
Yes, please. Go
Hi,
ahead.
this is Guang Wei from Almond Board of California. Thank you for putting together this report. We're waiting for, and then good to hold until you guys have thoroughly analyzed the data. I have a couple of questions about the feeding trial. The first of all, the gas measurement, those emission measurement, is a one-time measurement or just a whole day measurement? And then second question about the feeding trial, between feeding period for those their cross feeding, like any washing out period? For you have 3 groups, right? You switch that diet. Is there any washing period, 2 weeks washing period between the feeding periods?
Afroza, do you want to take that?
Yes. So I guess the first question that you asked,
The emission measurement is one day per, once, or if it's just once a day, at what time you measure after feeding?
So basically, they had free access. That means it's not a specific time point. So they had access for 24 hours, but
okay.
we set the system so that like after 3 hours later, so they had to come again. So I know what you are trying to point. Maybe after feeding, they might have more methane, but we try to capture the diurnal pattern or all the days emission. So no specific time point.
So those are by voluntary walk-in to measurement, is not by control measurement.
Sorry.
It's basically the cow voluntary walk to the
Yes,
instrument to
yes,
measure.
yes. They visited the system voluntarily. Yes.
Okay.
And the second question, I guess, was, well, there was any washout period, right?
Yeah.
So in our study, we did not followed any specific assigned like washout periods, but as we had they were visiting the system for 4 weeks, and then we might expect that by that time they will have some points after 1 week or 2 week. They might have like the natural washout in their system. I know it might be great if we had that set up for the like extra washout period, but we did not do that here.
Yeah, I think it's a balancing act. Do you want to feed the treatment for a longer period of time, or do you want to have the all animals go into control and then you feed the treatment for a shorter period of time? So we opted for having the treatment for a longer period of time so that they have time
Yes.
to adjust and all that. So yeah, it's a balancing act. It just depends on the preference. Sometimes you want to have I mean, if it is something that stays much longer, then you probably want to have to go back to the baseline and then come back again. But I think in this case, we felt that it's better to have a longer period of time that the animals are on the specific treatment. That way, when we measure the emissions at the end of each period, then we get a more representative sampling than having to limit it only to 2 weeks of the treatment if we had to do the washout period.
Thank you. So others, about you guys probably have experience about this. What's a typical methane emission rate or over time, over a day after feeding? So this is probably have something, for the future design, you might have tied it with this. I mean, after digested, they emit the methane whole day or just for certain couple hours after or few hours after intake of the food?
Yeah, so the animals were visiting the green feed multiple times a day. So some of it could be just after, and some of it will be much later after they've been fed. A lot of the animals actually like to go to the green feed, and we had to space it out. So if an animal comes in and visits the green feed, they are not allowed to come back to it for another 3 hours. So the animals they know that they will wait for the 3 hours and then they come back and then they have their measurement and then another 3 hours and something like so. We had that constraints and so the animals they train very well and they understand what's happening. So they like to visit it and they come back over and over again. So we usually get a good amount of visitation to the green feed. So it's not skewed to one time or another.
Okay, so in the early the literature review, it mentioned about the red algae have 90% reduction. Was that being proved in a feeding study?
Yes. We've done this work in 2018, where in dairy cattle we've seen about 67% reduction, but that is because we're using a different type of seaweed. But in beef cattle, the following year we saw about 80% reduction.
So in vitro is 90%, but in vivo is 80%. Yeah.
In dairy was 67%, in beef was 80%, but that's just because of the difference in species and difference in bromoform concentration. We just published a paper when we did a meta-analysis of all the work that has been done so far around the world. And what we see is that if you use sort of the midway, which is about 28 milligrams per kilogram of dry matter of bromoform, then you would expect about 50% reduction in methane emissions. So this is across about 11 studies that have been done around the world.
And Gang Wei, to your point, I think you're asking is there a correlation between what we see in vitro and in vivo? I think that
yes.
And it's pretty consistent. So when we see something in vitro, it's very likely that we see a similar level in vivo. So that's the reason why that turns out to be a pretty good way to test different combinations. I think that was your question, right?
Yeah, because
Yeah, I
you know,
think-
like even this one, because previously we noticed the in vitro study there is over 90% reduction, and then even in your second in vitro study still have over 30%, but in the feeding study we don't see any,
Yeah.
very little reduction. So that's why I'm trying to understand what happened. What's the correlation.
Yeah.
And the other thing, maybe the question to Hamas and then Rui Hong, I noticed that, in the early report, the ethanol concentration for this from this fermented whole is over 0.1 something percent, I suppose. And then in the feed use for the study, it's like a 0.04. There any difference? Would that contribute to the difference?
Oh, hi,
Yeah.
Gong. I could answer first, then I'll have Hamid to chime in. And the answer to your question, I think possibly, and I would think yes. And so from our research experiments so far, so we have tested different varieties of almond hulls. Certain hulls have higher sugar content than the others. And so the hulls that we showed, the hulls that showed the higher methane reduction in our lab experiments tend to have higher content of sugar, and compare with the hulls that have less methane reduction. And so as you point out, the hulls used for animal study for this wine have lower sugar content. So because of the hulls that came from a vendor. I think there's more to do there. Yeah, more research to be done, and in that regard, yeah.
So that's the feed, the almond whole feed to the study is a prime whole, or they didn't specify. I guess it's just mixture, huh?
It's a mixture, and the vendor or the supplier could not tell it's a large scale, and just almond hulls delivery to dairies, and so it's a mixed variety.
So
And
sugar content you measured was much lower, because I didn't capture that data. Composition for the feed, the almond whole used for the feeding study was
has a lower amount of sugar.
How much was
about 17? I think adding the sugars
Oh,
together.
that's half
17%. Yeah.
Oh, okay, half
An
lower.
early study we did with Almond Board, the funding, we used the Independence, and Independence has the most sugar, one higher sugar content, and faster and more fermentation with the yeast, right? Yeast eat sugar, and so that's now later we also tested nonpareil, and but then with this latest study where we show the animal study data, and so it's a mixed ration, mixed whole, so we don't know what variety. Yeah, we just
Can from the composition analysis, I know it's 17% sugar. What's the crude fiber content?
How many?
What's the crude fiber content or NDF?
I believe Feroza had a table.
I didn't capture it. I just wonder if for this
I wonder
maybe,
what was the
what's a grade of almond whole? If it's prime whole or okay. This doesn't accrue. I want to look at for crude fiber, but okay. No, that's okay. 17% of sugar content is really low. It looks
right.
like
It's rather low.
Yeah, it's
more
probably
than we
lower
normally
grade.
see. Yeah.
Let me jump in here real quick. I know Paul has a question, but I think this is really important. And Gangwei, you really bring up a really, really good point. And what I want to kind of emphasize here is really that we have funding from CDFA and CDRF to actually generate chemical compositions of some of our byproducts, and that includes almond hulls, fermented, unfermented, because what we do really miss is really an understanding of what are the chemical components in all these really complex mixtures that really result in lower or less, in the different levels of reductions. So this is kind of something we are, besides testing the very specific byproducts, we also try to build a knowledge base on very specific chemical components, because all of those can vary, as you know pretty well, not only between different varieties, but also depending on the season of when you harvest things and how you pre-ferment it. So I hope that we can add additional information that will allow everyone involved in this really to evaluate really how can we go towards a more formulated feed that really facilitates methane reduction. So I hope this kind of clarifies the complexity and also the opportunity we have right now with additional funding we have from people actually on the call here.
I'm so glad we have the same path, the same path. So, I really think maybe look, maybe based on the composition of the fermented hull used in the previous in vitro study, maybe trying to come up some combination of lactic acid and ethanol and acetic acid, maybe to do it, mix with some other in vitro study to see, what's the mechanism behind this.
So the idea, what we have, and sorry, Paul, for not letting you speak, but I think this is just, so the idea is that we built that database, and hopefully we can build an algorithm. We don't have funding for this right now, but the idea is right now there's very little coherent or data that we can compare. It's all over the place, different in vitro systems, different chemical composition that have been analyzed. So what we try to do with funding specifically from CDFA and CDRF is to build a database very specific on particular chemical components, but then also on how this affects rumen fermentation. So we hope to build a database that we then can actually go into and hopefully see some variation, or so see some correlation between the different components. So I agree with you what you're saying. It would be good to do these combinations, but the problem is they're limitless, indefinite ways of combinations. So we need to narrow that down a little bit to make sure that we actually have a reasonably good success.
Cool. Thank you. Sorry to take it so long. Yeah.
Thank you, Guangwei. And Paul, before I go to you, and there is a question from Jennifer that has been waiting for a long time. So can I just ask that quick question? It's I'm reading the question, and the 3 dyes that were formulated, and were there any differences in the chemical composition, and what percentage ration was almond hulls in the control? So maybe what percentage of ration of almond hulls in the control used in animal study? That's an important question.
Yeah, so the ration we used, they were isonitrogenous and isocaloric. That means maybe you see the different components here, but chemically they were similar. So they were not having any, we don't expect any differences based on their composition and percentage too.
Okay, thank you. And Paul, yeah, please.
Yep, sure. And I appreciate Matthias's conversation that he was just having on the variability. in answering the previous question. And that was what I was going to bring and appreciate that you guys did look at that variability. You brought in the variability from the DePeter study from 2020. And this is something we see on farm. Almond hulls vary from truckload to truckload. We have to deal with that in our feed rations. And that's an important component of this work. And I'm glad you guys did, that you didn't cherry pick, the very best almond hulls to run the study with. You actually took a commercial load and you got what you got, like we do on farm. And that's an important piece of this work, dealing with that variation and how do you deal with it and what results do you get when you're dealing with that variation is an important piece of that. And I'm glad you guys took a look at that and gave it some thought. And as Matthias was just, kind of going into that in detail, that's an important thing to continue to consider going forward.
Okay, well, thank you, Paul.
Thank you.
Okay, good. So we are closing into the end of time here. And are there other questions from the audience? If not, then I would like to thank you. And also, there's a lot more to do. And I think we are still in the initial stage, and we're hoping we do have a project going, and there's a lot more we could do, and with the more funding and the collaborations of partners, and including fermentation processes and different materials and different conditions, even scale up onto the farm conditions. I think there's more to do. And but I wanted to have Matthias, Mies, do you have final words to share?
I just want to say thank you. This has been really a pleasure to be involved in this and to work on this trial. And we hope that this is going to be useful for dairy farmers in California.
Yeah, I just want to second, yes, I think this, thanks for attending that workshop, providing funding, and I think there's a lot of more work to do, but I think this is a great team, and we do it because there's a real need, and I think it's really something where I feel deeply engaged about, and also really see that there is engagement from everyone involved, farmers and producers, and it's great. It's really good to see that everyone pulling in the same direction. That's great.
Great, thank you. And I would like to ask you for a favor and do a mini survey to help us, to get more of your thoughts. And there is a link in the chat. And if you can click that chat, and then it's just a few questions. If you can quickly answer, that will be very helpful. And so we are planning to follow up after the workshop. We do have the recording. And so I will be happy to share with you the recording and also the slides. And also we're preparing a research summary to share with you as well. So we'll give them a couple minutes and then we will wrap up and then conclude.